Good afternoon. My name is Ryan Urbanowitz. I'm from the University of Pennsylvania, and I'm presenting this research that was also developed with Satvik Desiragi, the first author. Our project's called RARE, Evolutionary Feature Engineering for Rare Variant Bin Discovery. So I'm coming at evolutionary algorithms from the perspective of biomedical data analysis, and there we usually have this potentially predictive pool of variables of interest, and we're trying to connect them with a disease phenotype of interest. And these relationships between predictive variables and outcome can be very simple, or they can be complex, such as the case in epistasis, where you have multiple variables that collectively, when you consider them together, are more informative of disease outcome than if you look at them individually. Feature engineering in predictive modeling is an important part of predictive modeling. So here's a typical pipeline for machine learning modeling analysis, starting from raw data and hopefully going to new knowledge. You would start with pre-processing, then have some sort of feature processing, modeling, and post-analysis. It would look something like this. And under feature processing, one of the components is this idea of feature engineering. And this is basically where you use domain knowledge to extract features from the raw data. So you use your understanding of the problem to try and create new, more informative features. And this is similar to feature construction, where you transform a given set of input features to generate a new set of more powerful features that are hopefully more important for making, or more useful for making informative predictions. So a little bit about rare variants, which is another part of this project. So what I mean by this is just features where certain states of that feature have a very rare occurrence. So understanding the relationship of that rare occurrence and an outcome can be very difficult. And in genetics, we have common and rare variants. So common variants would be, so every row here is an individual and every column is a different genetic loci of interest. And so here we have loci that occur pretty commonly within a population, but then you have rare variants that only occur in maybe one or a small subset of people that you've sampled. And rare variants lead to a number of challenges in statistical analyses and machine learning. Primarily, they limit our detection power, our ability to detect their effects on outcome. But also because they occur in such hard numbers, people have largely only looked at simple univariate effects of these rare variations with outcome and haven't looked at more complex effects like epistasis, one of the things we're considering in this study. Moving on, binning is a sort of feature engineering and is also the topic of today's talk. So binning is where you lump multiple categorical variables together into a single category. And generally you do this by applying some sort of domain or expert knowledge to engineer new categories and features that better represent the structure of your data, and in this case, better capture underlying associations. So the idea here is if we take a bunch of rare features and put them together in a bin that is now a new individual feature, that will have more power to detect their association by lumping them or summing them together. Traditional genetic studies have used binning, also known as burden testing. And again, here we're combining all rare variants within a single genetic region into a single bin, a single new variable. And then we'll evaluate that bin or that new variable with a simple univariate test of association with our outcome of interest. So here we can see a couple examples of genes, and binning is taking place by basically making a window across the spatial genomic region, and then summing up how many rare variants occurred within each region, and then looking to see if those bins or those summed counts of rare variants within a region are in itself informative of outcome. However, these methods rely on big assumptions, right? Like that these spatial regions are in fact capturing the best set of rare variants to be informative. And again, these existing methods haven't looked at interactions between rare variant bins and other common variants or other rare variant bins. The most cutting edge rare variant binning strategy in bioinformatics right now is probably BioBin. And BioBin just takes what I mentioned a second ago a little bit further, and beyond just looking at genomic windows or windows across a gene, it also allows you to look at a hierarchy of biological information to define candidate rare variant bins for further downstream testing. However, even this has a few major drawbacks. The first with this idea of rare variant binning, at least in genetics, is that variants can have different directionalities. So when you bin some rare variants together, some of them might have a protective effect of health and others might have a harmful effect. And when you put them together within the same bin, they can wash each other out and make it harder to see their relationship. These spatial boundaries also is a second problem, right? That's a big assumption. If those bins don't properly capture the informative rare variants that work together, then it's not going to work as well. And further, again, only univariate associations between bins and outcome have been considered. So that brings us to today's idea. We're presenting RARE, which stands for Relevant Association Rare Variant Bin Evolver. It's basically an evolutionary algorithm or a genetic algorithm that constructs bins of features that have rare states in an attempt to optimize bin association with outcome. Here's a schematic of the RARE algorithm. Again, it's a pretty basic genetic algorithm based, where we take our data set. We first identify which variables are rare, which are common. We do some sort of bin initialization. In this case, in this study, we're using either random initialization or we're simulating some prior expert knowledge that points us in the right direction of what might be a fairly useful bin. And then we're going to see if it can evolve further beyond what's given to it and find a more optimal bin. Then we have the standard, a pretty standard evolutionary cycle where we have our bin population. We evaluate the fitness of every bin, and we are testing two different evaluation metrics. One is a univariate test, the chi-square, and another is a relief-based feature importance algorithm that can detect not only univariate effects, but epistatic interactions. Once evaluation is complete, we apply our genetic operators, including parent selection, crossover, and mutation. And then this cycle is continued. And we're using elitism to preserve, in this case, 40% of the bins every cycle. And what we get out of this is a final population of bins that includes, so every bin is a candidate, something that might have a strong association with outcome that we might want to examine further. So we're asking 4 research questions in this study. The first, simply, can RARE adapt informative bins that have simple univariate effects from scratch? So starting from randomly initialized bins. Second, what are the trade-offs between fixed and adaptable bin sizes? Third, does expert knowledge bin initialization improve bin discovery? Does it do it faster, more effectively? 4, can RARE correctly identify bins that epistatically interact with some other holdout common variant? So for the first time, we're looking at rare variant bin discovery in the context of not just univariate effects, but epistatic interactions. A little bit more on bin initialization. As I mentioned, we're using a random initialization as well as expert knowledge initialization. And the way we simulated expert knowledge in this case was that we started our bins with 50 to 80% of the predictive variants, and as well as at least 4 non-predictive ones. A note that whenever a bin is discovered, or whenever we have a candidate bin, it represents a new constructed feature. And we basically sum the number of times that a rare variant occurred within a given bin, and that value becomes the new value of the constructed feature. Here's just another look at the evolutionary cycle. We start with bin fitness evaluation with either the chi-square or multi-surf. We are using elitism. Then our genetic operators are applied, and then we combine our elite bins with the newly discovered bins to create our next generation. And what we get out of RARE is, again, a population of candidate rare variant bins. But in this study, for simplicity, we're just going to focus on what was the best bin found in our population each time we run the algorithm. Here are the hyperparameters that we use in this study for RARE. Pretty simple, population size of 50, 3,000 evolutionary cycles, 40% elitism, 80% crossover, and 10% mutation. As I mentioned, we used 2 strategies for fitness evaluation that we tested. One was simple univariate scoring with the chi-square test. This is only designed to detect univariate associations, and it will not capture complex interactions like epistatic interactions. And it's very computationally efficient. Another strategy that we focused on in this study is using relief-based scoring. Here's a citation for relief-based methods down below. In particular, we use something called the multi-surf algorithm. It's designed to capture both univariate and univariate effects and multivariate interactions. It is significantly more computationally expensive. So in this study, for just simplicity, we always evaluated our bins using only half of the instances in our simulated data sets. For our experimental evaluation, we actually in this study also developed 2 different rare variant data simulators. We needed a way to simulate these scenarios of rare variant associations. You can see the paper for details, but one of these rare variant simulators creates univariate association bins, or bins with rare variants that have an association with outcome, and another simulator that creates rare variant bins that have an epistatic interaction with some other common variant that we've included in the simulated data set. Our experimental setup was designed to target our 4 research questions. So first, we looked at univariate binning, where we had different degrees of noise in our simulated data sets, because in real world problems like bioinformatics, the signal is very, very noisy, or it can be very, very noisy. Then we had data sets where we conducted univariate binning with different configurations of the rare algorithm as we were trying it out and testing out different ways to actually implement it. Then we conducted univariate binning, looking at random bin initialization versus that expert knowledge initialization I alluded to. And lastly, we conducted epistatic binning. Here are the data sets that we simulated in this study. We actually used 30 replicates of each for the analysis. Our first 4 target our first question, so we have different degrees of noise here. We are evaluating fitness with our multi-surf algorithm, and you can see we have 50 total rare variants, only 10 of which are predictive. Then we have, and so this last one with 50% noise, that actually equates to, it's just a noisy data set, so this serves as our negative control. Then we have a univariate data set where we're applying the chi-square test just to see how that performs in contrast with multi-surf. Then we're using, we're conducting analysis with constant bin size to see if the algorithm works, how much better the algorithm works when we know the approximate size that the bin should be. Then we used expert knowledge in a test. And lastly, we generated epistatic interaction data sets to see if using either our univariate metric or our multi-surf metric, the rare could actually detect, could identify bins that had an interaction effect with some other common variant. So here are our results. First, here's our first set of 4 data sets with univariate associations. Here we can see the increasing degree of noise, where our negative control is here at the bottom. We show the correct percentage of rare variants that were included in our top bin over the 30 replicates. These are averaged over 30 replicates, all these results. We also show the incorrect percent. So these are rare variants that shouldn't have been in an optimal bin that ended up in our best bin. Here we have our optimal chi-square. So if we just took our, if we just evaluated the chi-square test on what would have been the optimal bin, given this is a simulated data set and we know what the true best bin was, this is the ideal chi-square score that you would get. And then this is the chi-square that we obtained in our analysis running rare on the data set without knowledge of what the true pattern was. And as you can see, we're getting pretty comparable chi-square scores. Obviously, the optimal is a little bit better in each, but we're doing a really good job. And as our noise is increasing, you can see that the chi-square association is dropping as expected. But what's good here is that we're basically shown that rare can identify from scratch these rare variant bins that we've simulated with univariate effect. Next, we're looking at some different configurations of the rare algorithm. So first here, we have multi-surf as the evaluation metric versus univariate. And so this is actually the same as data set 1. And so you can see the values are the same. And when we used our chi-square test as the fitness metric, it was actually able to very easily identify the perfect optimal bins. This is likely due to the fact that when we ran multi-surf, we only trained it on half of the training data, while chi-square test got to use all of it. So that explains that difference, but it also shows that both algorithms are able to identify these rare variant bins from scratch using either metric. Further, when we tested a constant bin size of 10, which is would be the ideal bin size here, what we found is that we could get slightly better bins than we had gotten without fixing the bin size. And this is also somewhat expected and just demonstrates that if you have a pretty good idea of what your bin size should be, then using that information is certainly useful. Moving on to our next question, where we are asking about expert knowledge and whether it can help the algorithm find a solution faster. Here we have random initialization versus using our partial expert knowledge. So we're giving it that 50 to 80% of the true variables in initialized bins. And you can see that it's doing a slightly better job of finding the ideal bins. And notably, it was able to find much better bins much more quickly with a far fewer number of evolutionary cycles than without expert knowledge. So again, this is somewhat expected, but it's a good proof of demonstration that rare can work effectively either on its own, but you can expect it to work better if you give it a little bit of expert knowledge and point it in the right direction, as is the case with most evolutionary algorithms. Lastly, when we looked at our epistatic associations, we were able to demonstrate that rare similarly could find epistatic interactions between rare variant bins and a common variant. So in other words, it was able to discover a rare variant bin that had an interaction with some other common variant in the data set. And so this was very promising. Notably, whenever we used multi-surf to evaluate our algorithms, the runtime was significantly longer because there's a lot going on with rare in searching for both univariate and epistatic effects. And so one of the things we're doing in the future is figuring out how to scale this up. So conclusions. Our simulations show that rare reliably constructs bins with univariate association with outcome, as well as epistatic interactions with outcome. However, the second one can be a little bit more computationally expensive. Rare can discover informative bins from scratch. Combined with expert knowledge, Rare can more rapidly and accurately identify informative bins. Rare offers a promising data-driven approach to rare variant binning that's unbound by the assumptions of the way things are currently done in genetics using just expert knowledge to define bins. And in this work, we've also presented 2 rare variant bin simulators that can be applied in the future to test new strategies for rare variant binning methods. Applications and ongoing research. Rare could be applied to bin features in any kind of data set containing features with rare states. It constructs bins of rare genetic variants to improve predictions of disease phenotypes and elucidate complex interactions. One of our projects that we're looking to apply this to is a study of donor-recipient donor-recipient matching and the idea of graft failure. When you have a donor organ, what are the chances that it will not stick? And so we're basically applying this to that study where there's a lot of rare amino acid mismatch states. Future work, we want to explore intelligent instance subsampling methods to overcome some of the computational requirements of relief-based scoring. We think we have some strategies that might help with this. We also want to conduct bin evaluation based on different metrics other than just the chi-square and multi-surf, and we have some work going on there. We also want to evaluate and expand rare scalability, so apply it to much larger data sets. This is really just proof of principle. And ultimately to develop open source scikit-learn package for users to easily run and apply rare. With that, I want to thank the primary author, Satvik Desiraju. He's actually a high school student, extremely talented, did the bulk of the groundwork for this project. And our collaborators, Lauren Granger from Tulane University and Malik Kalmoun from my institution here at Penn, as well as Robert Zhang, an undergraduate who helped make this code available and get it out there. And lastly, funding from the National Institutes of Health. And with that, thank you very much.